Thank you, everyone. I very much appreciate being included in this talk. My name is N Schairer, and like they described, I'm the co-director of IBD at the Henry Ford IBD Center. I'm located in the United States, and I'm talking today about emerging technologies in IBD. Specifically, can we do a better job diagnosing and assessing the IBD? I have no disclosures at this time. We start with an expression, and it's that it's no use running quickly down the wrong path. So in IBD, the most important question is, does this person actually have IBD? Followed up by, do they have Crohn's disease or ulcerative colitis? And traditionally, clinicians have done a very good job at making this decision. But it unfortunately occurs typically after a bout of severe inflammation leading to symptoms. And as described in earlier talks today, most patients have had a prodrome or preclinical period where there was inflammation and there were symptoms. They were too mild to be detected. So we've all had that patient who presented at a young age and went to surgery rapidly. It's not that they just got IBD and went for surgery, it's that they had months or years of untreated disease. And so we ask, can we do a better job identifying who is going to get Crohn's disease or ulcerative colitis early? This study looked to assess that by using biomarkers. And some of them we're familiar with. In terms of antibodies, we know the ASCA, the PANCA, anti-CBER, anti-flagellin antibodies. They also looked at over 1,000 protein markers, and those are included in a wide variety of things like innate immunity, complement cascades, glycosaminoglycan metabolism, and lysosomes. And from this collection, they pulled 51 biomarkers and analyzed them to see if they could predict who was going to get IBD. Now, in this area under the receiver operating curve, just a reminder, as we approach 1, that is significant, and as we approach 0.5, that's considered not significant. And at the 1 year, what they did was they looked at biomarkers and then found out who was diagnosed with Crohn's and went backwards and said, how far back were these biomarkers predictive? So at 1 year, the AUC was 0.85. They were able to predict Crohn's disease a year prior to diagnosis. But the methodology held up so that 5 years prior to diagnosis, the AUC was still 0.76. It was still significant, suggesting that up to 5 years prior to diagnosis, we have an inkling of who may develop Crohn's. They put ulcerative colitis patients in the same methodology and did not find significance, so it only works for Crohn's patients. So what's the significance of something like this, a panel of biomarkers? Well, I'm not going to suggest we should screen the entire population. That does not seem feasible for such a low incident disease. But if there's somebody who you suspect Crohn's disease in, and you've done the appropriate workup, the colonoscopies, et cetera, that have been described previously, and they've escaped diagnosis, tests like this may be able to risk stratify someone, identify someone high risk for Crohn's disease that we follow more aggressively, and we get objective markers over time so we can provide an earlier diagnosis. Another way to look at early diagnosis, as Shaji mentioned, is genetics. And starting in 2001, we identified the first gene associated with IBD, the NOD2. I was actually in medical school at that time, and I remember it well because it kept showing up on all my tests, and I couldn't figure out why. But since then, we've been doing genome-wide association studies, and we've identified over 200 single nucleotide polymorphisms associated with IBD. There's other ways to look at this. When we look at it from a genetic perspective, we begin to understand that perhaps IBD is not a single entity, or that ulcerative colitis and Crohn's disease are not the only two subclassifications available, but rather, is this a polygenetic disorder? Is it associated with multiple things like dysregulation of autophagy, phagocytosis, cell signaling, fibrosis, and other autoimmune conditions? And the genes seem to play a role in that. So the hope is that by looking at the genetic profile, we can start to identify genotypes, begin to predict phenotypes, and understand which medications are appropriate for an individual person. And this is the very definition of individualized therapy. The issue in 2021 is that so far we've identified 13% of Crohn's disease genes and 8% of ulcerative colitis genes that account for the variance. And honestly, I don't know where the denominator of that comes from, but it illustrates that we have an incomplete picture to be able to thoroughly understand the genes. We've also looked through next generation sequencing at whole exome sequencing and whole genome sequencing. And by focusing on whole exome sequencing, which is where we look at genes that are in charge of coding exomes or the protein coding portions, we can reduce the amount of our genetic material we have to look at down to about 1% of the total, and hopefully apply our resources there so it's higher yield research. Genes make a lot of sense to us, though. We understand the concept of having them, putting us at risk, and we understand mutations can put us at risk. But we also remember back from our training, penetrance, the fact that just having a gene or having a mutation is not enough in all scenarios to get the disease. So what goes into penetrance? Well, we understand now epigenetics, how, for instance, the cells around the cell in question can affect the genetic expression within that one cell. And also, can our environment play a role in which genes are expressed? So in IBD, 3 of the big areas of epigenetics that we're looking into are histone modification, DNA methylation, and non-coding RNAs. I borrowed this graphic because I really like it. And if you follow the green line from left to right, that is a strand of DNA. The DNA has wrapped around histones for the purpose of storage, and that makes it so that you cannot transcribe the DNA. The gene is functionally off. As the histone chemistry is modified, it may hold on to the DNA tighter, or it may allow the DNA to unwind, which makes it available for transcription. But just having the DNA not associated with a histone is not necessarily enough for transcription to occur. So another mechanism that affects gene regulation is DNA methylation. When the DNA is methylated, the transcription factors cannot bind. So again, that down-regulates the gene. And by demethylating the DNA, it allows the transcription factors to bind and create coding RNAs. I do appreciate that I've oversimplified this, but in the essence of time, I'm trying to express a complex concept in an easy way. The third thing I mentioned was non-coding RNAs. So when we have the mucosa of the GI tract and becomes inflamed, some of those products of inflammation are created by messenger RNAs leading to proteins. But another type of RNA is called microRNA. And microRNAs are very small by comparison to messenger RNAs. They've been implicated in things like intestinal permeability, inflammation, fibrostenosis, and apoptosis. And what happens is the microRNAs can bind to a messenger RNA. So if there's an area for transcription and the microRNA has bound to that area, transcription does not occur, and that can downregulate. Another example would be if the mRNA is set to be destroyed and the products are coming by to interact that begin that process, the microRNA can be in the place where it inhibits the degradation of the messenger RNA, allowing the messenger RNA to persevere and code further inflammatory processes or proteins. So microRNAs are implicated with inflammation in the GI tract. And certain studies have shown that it's not just 1 or 2 microRNAs, there are many different types, and they can be associated with the diagnosis of IBD, diagnosis of Crohn's disease, and diagnosis of ulcerative colitis. So this is encouraging, this is interesting. But this comes from tissue samples. And if I'm trying to create a useful diagnostic screening or surveillance test, then I'm looking for something that is inexpensive, easy to perform, safe, and highly reproducible. And in this case, if I have to go get tissue, if I have to do a colonoscopy before I can provide a diagnosis, well, then I might as well just do a colonoscopy and provide a diagnosis the standard way. So the next question becomes, can I look at these same microRNAs in the serum? And they can be detected there, but they're not a reliable indicator of inflammation or even the diagnosis. So they can't be used with those particular mRNAs. The work of other researchers like Iborra and Christensen have shown that they can detect circulating microRNAs in the blood, and that these do correlate with active Crohn's or active IBD. In the case of Chen, microRNA 146B5P has a very high sensitivity and specificity of 85% each. These studies are all small, and they need to be reproduced multiple times on larger scales. But it's interesting that we may be able to diagnose or assess IBD with other biomarkers, such as microRNAs in the future. Another aspect of epigenetics is the microbiome, something that we have a lot of interest in, but have always had a hard time connecting directly to IBD. Can genes affect our microbiome, and can the microbiome affect the expression of genes? Well, we know that the NOD2 codes for our response to commensal bacteria, so clearly the genes play a role in which bacteria are present. But to look the other way, can the microbiome affect our gene expression, we don't have direct evidence of that. We have things that make us question. For instance, we know that the locations of the worst inflammation in people, things like the terminal ileum and the rectum, tend to be associated with more stasis than the rest of their respective organs. And we know that if we have severe proctitis or severe perianal disease and we divert the stool through an ostomy, things tend to get better. And if we reverse the ostomy, the inflammation tends to get worse. That's not a definitive, obvious statement, yes, the microbiome affects it. It could be the microbiome, but it could also be nutrition from the stool going past. It could be the products of digestion like bile salts. We're not sure, but it begs the question. And the third indirect piece of evidence that makes us question, should we look at the microbiome as affecting epigenetics, is that we know people who had antibiotics within the first year of life are more likely to get Crohn's disease. Now, I don't want anyone to leave this and say my child shouldn't have antibiotics because I don't want them to get Crohn's. The incident rate is still very small, but we don't know what plays what role in which part. Is it that having dysregulation of your immune system because of a genetic predisposition to, for instance, Crohn's disease, causes you to be more likely to get otitis media? Or does the antibiotic itself affect expression? Does the antibiotic affect the microbiome and the microbiome affect expression? But the point is that more and more researchers are looking to the microbiome to see their role in IBD. And I want to take a moment to pause and talk about what we mean when we say microbiome, because I think most of us focus on bacteria, right? Bacteria are easy. We can see them with a microscope. We can get a culture and know what bacteria are in there. That makes sense to us. But when I was in school in the 80s and 90s, we were taught that there were 5 kingdoms of life. And over the past 20, 30 years, we've learned that we can classify life many different ways, and now there are 7 kingdoms of life. One of them is called archaea. Archaea are single-celled organisms that live in very inhospitable environments, places like lava vents in the bottom of the ocean floor. So why is this guy talking about lava single-celled organisms in IBD? It's because Methanobacterium specializes in living in highly anaerobic environments, and that we have documented this in the presence of people with IBD, in the healthy population, people who eat salted meats, people who eat pork. The point is that archaea is commonly in our microbiome, but because it's so highly anaerobic, if we were to culture for it, we're not going to find it. And so we have to know that it's there to look for it. And that's why when we talk about microbiomes and we talk about bacteria, we have to acknowledge other things like fungi that are supposed to be there, or viruses that are supposed to be there. And in particular, bacteriophages. These are viruses that prey upon bacteria. And so when we study the microbiome, appreciate that it's a larger conversation, just do we have good bacteria or bad bacteria? To date, the question of the microbiome in IBD has largely focused around probiotics. And really the only 2 aspects we found it to be helpful are VSL number 3 for bacterial pouchitis and VSL number 3 for mild ulcerative colitis. But we don't have any data that suggests that this is going to be useful for severe disease. And in my own experience, in both those cases, the response has not been durable. We often end up switching to medication after a period of months to years. So probiotics, prebiotics, symbiotics are not widely effective for the treatment of disease, so there is interest in learning more. And likewise, we use FMT, but really the only place we have excellent data for it is in the role of treating refractory C. diff. We're looking to see if changing that microbiome can affect the IBD therapies, but to date, it has not been something that, again, has been shown to be standard of care. So for instance, I had one patient in my clinic who had severe ulcerative colitis and went for FMT. She clinically improved, but it was not durable. And ultimately she ended up going for surgery. That's not to say that FMT can't work. It's probably just patient selection. We probably sent somebody who already was in a bad situation. But people are looking at microbiomes as a way to affect Crohn's disease and ulcerative colitis on a deeper level. So I'm going to wrap up today posing questions for the future. These are emerging technologies. And what do they offer us? Can they allow us to find an earlier diagnosis for somebody and allow us a correct diagnosis of Crohn's disease and ulcerative colitis? Can we understand the genotype and the phenotype to the point that we can pick a medication tailored to the individual rather than just saying, you must use A before you use B, before you use C? Can we just go straight to C because we know that's the mechanism that's going to work for you? And while I didn't have time to discuss it, we're also looking into redefining what the appropriate endpoints are. Should it just be deep remission? Is there a role for histological healing, which I think the data has kind of shied away from? And all of this dances around the most important issue in IBD. Can we find a cure for IBD? Because once that happens, the rest of these conversations become a moot point. So I want to end my talk here. I sincerely appreciate being included in this conference. I look forward to seeing it grow over the years, and I've been listening for the past few hours and found it very educational. So thank you very much for including me.